اوكي هنبدأ.
تفضل.
Okay. Assalamu alaikum everyone. Hope you're doing well, and it's a great pleasure to be here with you today to discuss a very interesting topic: computational biology tools, to use or not to use, or to trust or not to trust. So I'm going to talk briefly about computational biology tools, their applications, and then I'm going to after that, I'm going to elaborate on a specific application, which is the generation of smart drugs against hepatitis C virus, followed by explaining a new approach in drug design. And finally, I will tell you my story with the computational biology tools and how it really helped me throughout my postgraduate studies. So let's start. Okay, introduction to computational biology tools. Actually, computational biology tools dates back to the early 70s. Or let's say precisely, the bioinformatics dates back to the 1970s, early 1970s. And it all began when the biological researchers revisited the idea of using computers in order to evaluate and compare large biological data sets, so as to make it easier for them to compare one biological data set to another. For instance, if we're talking about the genome and the genetic sequence, and so on and so forth. Also, among the important applications of the computational biology or the bioinformatics is modeling and also analyzing biological data, where bioinformatics simply help to simulate certain conditions or even help obtain a predicted tertiary structure of a protein with an unknown structure. This is called actually the homology modeling, which is predicting the tertiary structure of a protein with an unknown structure. Also, it enables scientists to compare between different species in regard to their genomes, specific genes, and also there is something very important, and actually many research has done extensively on it, which is the single nucleotide polymorphism, or SNPs. What a SNP actually is a DNA sequence variation. It occurs when a single nucleotide like A, T, C, or G in the genome differs between members of biological species. Okay, for instance, okay, computational biology tools are important in studying the SNPs, but what is the significance of SNPs in general? Well, SNPs, or single nucleotide polymorphisms, are mainly important in the genome-wide association studies, the GWAS, or in personal medicine. For instance, some people respond to certain treatments differently than others, and this was found to be due to SNPs, or single nucleotide polymorphisms, in certain genes, and also in how it could affect, or the SNPs could affect, the response of individuals to pathogens and different diseases. Also, from the other uses of bioinformatics and computational biology tools is its essential role nowadays in drug discovery, as we will see later. As we can see from this slide, bioinformatics is considered an interdisciplinary science because it simply blends different fields, as we can see: computer science, statistics, genetics, biology, and so on. So it's an interdisciplinary science. Okay, of course, everything in life has advantages and disadvantages, and computational biology tools do also. So I'll start with the disadvantages. Actually, the computational biology tools, for example, we said before that they help in creating homology model, or predicting the structure of proteins of unknown structure. But up to now, it is still impossible to create a 100% accurate homology model of a protein structure of unknown structure. And another point actually, which might not be a disadvantage, but it's like a misperception, for instance, is that up to now there are some scientists or researchers that don't trust the results obtained from using computational biology tools. Although nowadays we need to trust these tools because they help a lot, as we will see. Another thing is that they need good analytical skills, which means that a researcher who is using the computational biology tools should be aware of what exactly does he want and the criteria he will base the results on, so as to optimally benefit from using computational biology tools. What about the advantages? Actually, there are several advantages. Computational biology tools simply cut it short. It decreases the time needed to undergo wet lab experiments, as we will see. So instead of spending years in a lab doing experiments, you could spend months. It also helps in simulating the biological processes, in addition to showing different binding modes of compounds when binding to target proteins, as also we are going to see later. So simply, it simulates real life inside the computer. For this reason, it helps the scientist at least predict what will happen when he does the experiment in the real life. So he could take certain precautions, for instance. Another thing, and as stated previously, it helps predict structures that are not there for certain proteins. Okay, so after giving a brief or a glimpse about computational biology tools, now we're going to start the second part, which is discussing a specific application that computational biology tools were used in. To date, actually, hepatitis C virus is still considered one of the major public health problems, infecting around 300 million patients worldwide. For this reason, it's considered a public health burden. And several drug classes were extensively researched upon, like polymerase and protease inhibitors, in addition to nucleoside analogs. And of course, we recently heard that one of the drugs that is considered the first all-oral interferon-free regimen was approved for treating chronic HCV. It was FDA approved, and I think you know it. It's sofosbuvir or Sovaldi. Actually, it is a second generation uridine nucleoside analog polymerase. But still, actually, although it's considered now like the golden drug for HCV, it is of a very high cost, and also there is still a need for interferon in some genotypes, like genotype 4. So it's still not yet the totally interferon-free based therapy. Hence, the anti-HCV drug discovery shouldn't stop yet. More efforts should be exerted to generate new drugs that could be of less cost. And also, it would be used for all genotypes without the need of interferon. So we need a drug that is of lower cost and that could help treat all genotypes without the need of interferon. It's somehow hard, but it's, who knows, maybe one day we will have one. So now I'm going to discuss a drug discovery process that involves an in silico phase, which means in silico means using computational biology tools. So nowadays, most of the drug discovery processes starts with an in silico phase or using computational biology tools, because as we said before, that it cuts it short, or it decreases the amount of time spent in the lab. So in general, if you would like to generate a drug, you should know your disease and your target protein. And why should we know our target protein based on the disease? Because we need to know what sort of drug do we want, and where exactly do we want this drug to bind? We want at the end a smart drug or a selective drug. And why do we want a selective drug? In order to decrease the side effects. So the first step in the in silico phase is the homology modeling. In condition that you don't know or there isn't yet a known structure for your target protein. So if you have a crystal structure for your target protein in the protein database, a PDB structure, then you don't need a homology model. But if there isn't, then you need a homology model if you want to pursue in silico studies. And actually, there are different homology modeling techniques or tools that can help construct a model of the target protein. And simply, it constructs a model of the target protein from its amino acid sequence and an experimental three-dimensional structure of a related homologous protein that acts as a template. So we have the sequence of the target protein and we have a template. And simply the template is what helps create the 3D structure at the end. So homology modeling mainly relies on using known protein structures that are likely to resemble the structure of the query sequence or the target sequence. And actually it yields an alignment that maps residues in the query sequence or the target sequence to residues in the template sequence. The quality of the model depends on different factors, such as there's several factors that actually affect the quality of the generated model, such as the quality of the sequence alignment and the template structure itself. Also, the homology modeling actually could be really complicated if there are alignment gaps or indels that are present in the target sequence and they are not in the template. It would make it more complicated or sophisticated. Also, as the sequence identity decreases between the target and the template, actually the quality of the model declines more and more. So this is for the homology modeling. An example actually of a protein that is really significant but didn't have a crystal structure up till now, until recently actually, when Kong et al., Kong and his research team were able to resolve the crystal structure of the core of envelope glycoprotein. There wasn't any crystal structure before that. It is actually the hepatitis C virus envelope glycoprotein. And it is an example, this figure shows an example of one of the homology models of hepatitis C virus envelope glycoprotein. And this model in specific was based on the sequence of the envelope glycoprotein of tick-borne encephalovirus. So from here we know that the template was the envelope glycoprotein of tick-borne encephalovirus. So this is just to show us an example of a homology model that was based on a template. But fortunately now we have part of the crystal structure of the HCV envelope glycoprotein, which could enable actually scientists and researchers nowadays to sort of develop the research done in order to create vaccines and drugs that target hepatitis C virus envelope glycoprotein. Okay, so the third step after the second step after the homology modeling is doing the virtual screening. So what does virtual screening mean? Virtual inside a computer and screening. So instead of screening millions of compounds in the lab, we do it virtually inside the computer in order to obtain certain hits that then we could take to the lab. So the second step is virtual screening, where we choose certain library of compounds that we prefer. We could even choose the natural compounds that are extracted from from herbs or so on. So this is followed by using programs like Autodoc in order to do the virtual screening runs against as much ligands as you want. So it really facilitates, like you could do screening against millions of compounds actually. So simply you can select the site in the target protein and create what is called a grid parameter file. So the grid parameter file simply is a box that includes the residues that you want to target. And how did you know these residues? You knew them, or you were able to identify them from previously published literature. You knew that these residues, for instance, or those residues are significant in the pathogenesis of the disease, so you want to target them. Then you select the library of ligands, and this is followed by starting the run and then analyzing the data. So imagine if you had to screen millions of ligands in the wet lab or using experimental techniques instead of virtually. How many years would you spend inside the lab? So the third step in the in silico phase would be the analysis. You need to analyze your results based on certain criteria of selection that you predetermine. So you will start analyzing the data and determining the virtual screening hits based on specific criteria of selection that you have in mind. And then you will see the number of strong contact points of the ligand with the protein, and also determine the free binding energy of the ligand when it binds to the protein. And also, according to the results, actually, you could categorize the ligands into those that could be used in fragment-based drug design or other drug design approaches. And it is all based on the binding, on how you feel they bind or how strongly they bind to the target protein. So to summarize the in silico phase, you have the Autodoc tools that help you view the 3D structure of the target protein, followed by selecting the specific residues that you would like to target based on previous literature, followed by defining the grid boxes in order to guide the virtual screening runs, and followed by preparing or defining the docking parameters, and then undergoing the virtual screening run, followed by viewing the results and analyzing them. So this is, in a nutshell, the summary of the in silico phase. As for the in vitro phase, which follows the in silico phase, in order to validate the data that was obtained or that were obtained from the in silico phase, we use several techniques. So here we are simply, as I said, validating the data obtained from the computational biology tool. So we use, actually, we can use one of two techniques, either the surface plasmon resonance or dual polarization interferometry. Sorry, here there's a typo. I think it's not SPI, it's SPR and DPI. So both of them actually are a lab on a chip platform. So we will see why lab on a chip or lab on a chip technology. We immobilize the protein or the target protein on the chip, and then we pass the selected ligands one by one in order to observe their binding behavior and determine their binding affinity and the change in response units. So yes, so passing the ligands one by one in order to determine the response units. And also accordingly, we categorize the ligands further. As we can see in this illustration, actually it shows us the, where's the pointer here? So this is the chip, and then we can see the immobilized target protein, and then we can see the ligands that bind. So every ligand that binds gives a change in the response units. Okay. This slide actually shows us the binding and dissociation of different ligands where, as we can see, some ligands have higher binding affinities, but their off rates are faster than others. For instance, let's compare this to this. This has higher binding affinity, or almost the same binding affinity that's like this ligand, but the dissociation rate of this ligand is much faster than this ligand. So actually, it all depends on what compound do you want. Would you like a compound that binds strongly but dissociates quickly, or has a slow off rate? It all depends on the criteria that you determine. Okay, so after doing the preliminary experimental validation, we could move a step forward and do the HCV infection inhibition assay using the HCV cell culture and pseudoparticles. And of course, if we want to see the effect of the drug on different HCV genotypes, we could use that as well in order to determine which genotypes does the drug affect. So now we finished the in vitro phase. So we are going to discuss drug design. And because all the processes that we discussed in the previous slides are all aimed at drug design, they're all aiming to design selective drugs that are of less side effects, that are smart. So let's discuss a little bit about drug design. Among the approaches that are used in drug design is fragment-based drug design, where you have a fragment and then you build on it. It's as simple as it is. From the name also, fragment-based drug design. You have a drug lead and you optimize it by adding certain pharmacophores or groups. The second approach actually is the new approach, which is called the selective high affinity ligand, the generation of selective high affinity ligands. So this is actually what we are going to discuss. Selective high affinity ligands, the generation of selective high affinity ligands is a novel approach that was used firstly against non-Hodgkin lymphoma, and then it was used also in certain diagnostics. So what are they? What are the selective high affinity ligands? They are synthetic compounds that are generated by the help of computational biology tools. They mimic monoclonal antibodies, but they are 1/50th the size of monoclonal antibodies. They are robust, so they could stand long shelf lives. And they were found to be of high therapeutic index, high specificity, and they could be easily incorporated. For instance, they could be used as drug carriers in diagnostics, they themselves as drugs, and they are easily generated when compared to monoclonal antibodies. Because monoclonal antibodies actually needs more expertise, whether using the hybridoma technique or the page display. So it's so different. Monoclonal antibodies could take months, but shells could take weeks. They could be monodentate, bidentate shells, tridentate, and even tetradentate. And as the number of ligands inside the shell increase from monodentate to tetradentate, actually the binding affinity increases from nanomolar to picomolar affinity. And this actually shows certain criteria of selection that should be considered when we choose certain ligands to be incorporated into the shell. So we simply need a ligand of free amino or carboxyl group in order to be able to link the two ligands or the three ligands together. So how do we link them? We will see later on. But for instance, this is a target protein, and we can find a ligand binding to one of the promising sites according to previous literature. Okay, but there is another promising site also in the back. So what if we want to hug or embrace this site? We want to to sort of cover the whole site in order to have an effective drug. We found that there's another ligand binding to the other site proximally to the first site. So this could lead to the conclusion that these two ligands actually could be linked together to form a bidentate shell. How do we link them? We link them using many polyethylene glycol moieties, as we can see there, the COOH and NH2, and also a lysine. So the many polyethylene glycol moiety actually helps us to control the length, of the shell, and the lysine gives the branching in order to mimic the monoclonal antibody. So we simply measure the distance computationally between the carboxyl group, for instance, of one ligand and the amino group of another. Like you can see here, the amino group of another. And based on the distance, then we know how many moieties of many polyethylene glycol we could use. So these are the two ligands that we saw binding to the target protein, and this is lysine together with many polyethylene glycol bound together. This part actually is bound to the solid resin, and then so the shells are generated using solid phase synthesis or ATMOS, and then it gives us a bidentate shell having the two ligands. Okay, so this was one of the new approaches of drug design, which is selective high affinity ligand. This is Shah. Okay, so now it's time to tell you my story with the computational biology tools, actually, and how they really helped me, and they were of great help throughout my postgraduate studies. What I did is that I started the in silico phase, and I determined the target protein, which was the hepatitis C virus envelope glycoprotein. And then we did a homology model based on certain templates. And this was followed by doing the virtual screening runs against the envelope glycoprotein. And then after viewing and observing and analyzing the virtual screening ligands, we determined the top virtual screening hits in order to take them to the next step, which is the in vitro phase or the experimental validation. So the primary experimental validation was done using surface plasmon resonance in order to test the ligands and see which really bound to the target protein. And this was followed by doing the HCV infection inhibition assay. So after all of this, actually we found that there was one ligand, T16, that was found to inhibit hepatitis C virus in a genotype-independent manner, which was really thrilling and exciting. It was found to inhibit genotypes 1A, 1B, 2A, 6A, 2B, 3, and 4. And this shows the IC50 values of T16. So as we can see from the story is that computational biology tools really help in the drug discovery process. So what is the postulated mechanism of action of such a drug lead or a drug, hopefully at the end? So of course, usually hepatitis C virus interacts with several host factors like the CD81 and the Claudin-1 and several other host receptors on the hepatocyte, leading to the propagation of the infection. Hopefully, when the shell binds to the envelope glycoproteins at specific residues, they will inhibit it from interacting with the host receptors. And accordingly, they will inhibit further progression of the disease and infection of hepatocytes. Another thing is that it was found that when the hepatitis C virus interacts with the host receptors via the envelope glycoprotein, this stimulates signal that actually triggers pro-inflammatory cytokines to be produced, which leads to the liver damage and eventually cirrhosis of the liver. So also having a shell that inhibits such an interaction will prevent the progression of the disease into liver cirrhosis and finally hepatocellular carcinoma. So this was the postulated mechanism of action. So in order to sum up everything I said so as not to get confused and in order to have our take-home messages with us, and in order to see how positive was the impact of computational biology tools and not to forget that, let's see what we discussed together in this presentation. We discussed what are in computational biology tools and how are they revolutionary and how do they facilitate several researches or research fields and help in let's say expediting them, such as drug discovery. We also discussed that the computational biology tools actually help decrease the amount of in vitro and in vivo experiments because it cuts it short simply, as we saw, that instead of testing millions of compounds, we test few compounds based on the analysis inside the computer. Also, we saw that it helps in predicting certain homology models or constructing homology models when we don't have a crystal structure for the protein that we want to target. And that they are used in fragment-based drug design and generation of selective high-affinity ligands. Also, we discussed the different techniques that could be used after the in silico phase in order to validate the data that were obtained from the computational biology tools, such as surface plasmon resonance, dual polarization interferometry, which are called lab-on-a-chip technologies. So finally, I ended with sort of a proof of concept that computational biology tools are really effective in the drug discovery process, especially when we had at the end a ligand, P16, that was found to inhibit the hepatitis C virus in a genotype-independent manner. So it was really exciting. Finally, I would like, after ending this presentation, to ask you, do you think now that you could trust the computational biology tools? Do you think that we should use it or we shouldn't use computational biology tools? Well, I hope that this part has a larger number compared to this box because computational biology tools should really be trustworthy now. I would like to thank you so much for listening, and if you needed any more elaboration or any questions, if you have any questions, please send me an email on reemalaby@gmail.com, and I would be more than happy to answer. Thank you so much.